Good morning, students. In this lecture, we will discuss about chromosomes, genes, alleles, phenotypes and genotypes, etc. So at first, we are discussing about chromosome. As you know, chromosomes are the carriers of genes, because genes are the specific segments of DNA, and DNA and histone proteins are two components of chromosomes. That's why genes are carried by chromosomes, and therefore chromosomes are known as carriers of genes, as well as it is known as vehicles of genes. Now, chromosomes are first studied by Walther Fleming in 1882. So it is first studied by Walther Fleming in 1882. Genes are the specific segments of DNA or nucleotide. You can say it is the specific sequence of nucleotides. Genes are also known as unit of heredity or inheritance, as it carries informations from parents to their offsprings or progenies. That's why genes are called as unit of heredity or inheritance. Again, you have studied before, alleles are the alternative forms of genes, because genes are always found in pair, and each alternative form of genes is known as allele. Suppose we are taking an example for tall plant. Here capital T are the set of genes. Here it is genes, and capital T and capital T are the alleles. So in this way, here these are the two alternative forms, and these are known as alleles, whereas it is a pair of genes. So genes are always found in pair, and each alternative form is known as alleles. Suppose here these are the two genes, means each one is the allele, and here these are the loci. Loci is the plural form of locus, means a specific position. Suppose these are the genes, and on each allele, the specific position has particular type of gene locate here. So here it is, suppose small b, it is capital B. Suppose here small g, here again small g. So, in this form, these are the loci present on the allele, and here these are specific genes. So, chromosomes contain DNA, and part of DNA are known as genes. Again, we have seen there are a number of organisms in which same number of chromosomes found. Suppose we are taking an example, tiger, lion, cat. The number of chromosomes, 38 chromosomes they contain. Again here 38 chromosomes, and cat also contain 38 chromosomes. Simply, different organisms contain different chromosome number. But there are a number of cases in which number of chromosomes are similar in different organisms. Here we are taking an example of tiger, lion and cat. All these three contain 38 chromosomes, means 19 pairs of chromosomes. 19 pairs. So here 19 pairs of chromosomes they have. There are a number of similarities in these three organisms, but a number of differences also. So what is the cause of these differences, these dissimilarities found in them? The secret is the genes. So genes which are found on the chromosomes are of different types, and due to different types of genes they contain, they have a number of dissimilarities. So, actually genes are the carrier of informations, genetic informations from one generation to the next generation. Or you can say, due to genes, different types of similarities and dissimilarities are carried from parents to their offsprings. So here genes are different on different chromosomes containing tiger, which are contained by tiger, lion and cat. That's why they are different in shape, size and different types of nature and behavior. Again, we can take an example of gorilla and potato. In these two organisms, number of chromosomes are 48. It means on the basis of chromosomes, gorilla and potato, which are two different types of organisms, gorilla belongs to animal kingdom, whereas potato belongs to plant kingdom. They have different types of genes on these chromosomes. That's why there are a number of differences between them. Now, there are two different types of genes, and there are two different types of genes as well as alleles. If the genes or alleles are superruling, then these are known as dominant. When the gene or the allele are submissive or subordinate, then these are known as recessive. So here dominant allele means super ruling allele are known as dominant alleles, whereas recessive alleles means submissive or subordinate alleles. So your super ruling means the allele which express itself in the organisms in F1 generation, then it is known as dominant alleles or super ruling alleles. Whereas those alleles which are suppressed by the dominant allele is known as submissive or subordinate. These are not expressed in the organisms or in F1 generation. There are two types of dominant alleles. These are homozygous dominant and second heterozygous dominant. Again, there are homozygous recessive. So these are the two different conditions found in the organisms. Here, if we take an example of tongue rolling, then capital R or capital R, small r, these are the homozygous and the second one is heterozygous dominant. Here capital R is the dominant allele which expresses itself in the organism, and small r is the recessive allele which can't express itself. So here if capital R type of genes present in the zygote, it means it is homozygous dominant. And if capital R small r type of genes present in the organism's zygote, then it is known as heterozygous dominant. It means if one dominant gene or allele is found in the zygote, then it expresses itself and the subordinate or submissive allele cannot express itself in the organism. Here, small r is the submissive or subordinate allele, whereas capital R is the super ruling allele. Again, in case of tongue rolling, if both the alleles are subordinate or submissive, then it is known as homozygous recessive, and here it is small r. So it can't express the rolling condition, but it is homozygous recessive condition, and it expresses only the non-rolling condition. So here tongue rolling and here tongue non-rolling. Non-rolling. Tongue rolling is a genetic inheritance in which a person makes the tongue U-shaped. If you make your tongue U-shaped, then it is tongue rolling, and if after making an effort you can't make your tongue U-shaped, then it is known as non-rolling. So most of the people in the world are tongue rollers, means they contain capital R or capital R small r type of alleles or genes in their cells on the chromosomes, on the particular chromosomes. So here these are the two conditions of the alleles. Now genome. Genome is the full complement of genes present in an organism. It means it is a full set of genes present on the chromosomes in an organism. Full complement of DNA, and DNA contains genes as well as intergenic regions. Genes and intergenic regions. Both. So genes and intergenic regions of DNA is known as full complement of DNA, and it is the genome. Or you can say full set of genes present in an organism. Present in an organism is known as genome. There are about 19,000 genes are present in human being. It means these 19,000 genes are collectively known as genome. So genome means full complement of DNA containing both genes and intergenic regions, or you can say it is the full set of genes present in an organism, and there are about 19,000 genes found in human beings. So the full set of these genes are known as genome. Here the first chromosome, first set of chromosome contain about 3,000 genes, whereas Y chromosome contains about 231 genes. So in this way, total number of genes means full set of genes present on the chromosomes are collectively known as genome. Now genotype. Genotype. Genotype means genetic makeup of an organism. Genetic makeup or genetic makeup of an organism, or you can say genetic combination of an organism is known as genotype. Means it is the set of genes present on the chromosomes. For example, here for tongue rolling, genotype is capital R or capital R small r or small r small r. So these are the three conditions. Here both the alleles are superruling. In the second case, one is superruling, second one is subordinate, and in third, both are subordinate alleles or genes, and therefore the set of these genes are known as genotype. It is the genetic makeup of an organism. Again, phenotype. Phenotype means any observable character which is controlled by genes are known as phenotype. Any observable character or feature controlled by genes are called phenotype. Means these are the features which are inherited from generation to generation. And these features, the appearance of these features are controlled by genes. So here example is tongue rolling, and non-rolling. So these are the two different types of phenotype which are controlled by genes, and therefore here tongue rolling and non-rolling are the two alternate features or phenotypes which are controlled by either capital R, small r, or small r, small r. So here these are the three genotypes for tongue rolling, and here tongue rolling and non-rolling, these are the two phenotypes which are controlled by the genes, and therefore these are genotype and phenotype. We are taking an example of inheritance of tongue rolling. Inheritance of tongue rolling means heredity of tongue rolling. Tongue rolling is a feature in which a person can make his or her tongue U-shaped. If they can make U-shaped tongue, then it is known as tongue rolling, and if they can't make U-shaped tongue, then it is called tongue non-rolling. So these are the alternative forms of the feature character. Tongue rollers are dominant, whereas non-rollers are recessive. Now, non-rolling are expressed by only homozygous recessive condition, whereas if any one or two superruling genes or alleles are found, then it can express the feature of tongue rolling. So here we are taking an example of tongue rolling. Suppose here squares shows males and circles shows females. Again, it is the solid structure, solid square, and again here it is solid circle. So solid circle and here solid square, and here it is hollow circle. So solid structures shows non-rolling, means it is non-roller, and hollow tongue roller. So here we are taking an example of inheritance of tongue rolling. The square shows the males and circle shows the females. Solid square shows non-roller, hollow circle tongue roller. Here solid square non-roller, hollow circle non-roller. Suppose here it is male and this one is female. You can say it is father and she is mother. After crossing, there are 1, 2, 3, 4, 5 children as progenies. Here again solid circle. It shows non-rolling daughter, whereas this hollow circle is the tongue rolling daughter. Here again tongue rolling, son, tongue rolling, daughter, and again tongue rolling daughter. In this way we can say there are only son, whereas 1, 2, 3, 4 daughters. Now here, which type of genes present in all these conditions, how can we determine? Here it is hollow circle, tongue roller, and these are here, this one and this one are the non-rollers. Non-rolling feature appears only when it is homozygous recessive condition. It means here the type of genes present is of small r, whereas here it is hollow circle, tongue roller. It means here capital R, small r, or capital R type of genes present. So here these are the two different types of conditions. Here small r means homozygous recessive condition for tongue rolling, and here this one is the first one is heterozygous dominant, and second one is homozygous dominant condition. Now when we study this pedigree chart means family chart, then we can say in the first one, which is non-rolling, small r, and from here small r comes. So it must means the parents must have small r, means subordinate allele in their cells. So from here small r comes, and again from mother small r comes. In the second case, tongue rolling case, here capital R from mother and small r from father comes, and therefore it is heterozygous dominant condition. In the third, which is the son, here capital R comes from mother and small r comes from father, and again it is heterozygous dominant condition. And in the next case, here it is daughter, tongue rolling daughter, and once again capital R, small r, means one superruling and second one is submissive genes or alleles present in them. And in the last, means fifth, again it is tongue rolling daughter. So capital R comes from mother and small r comes from father. In this way, tongue rolling and non-rolling characters can appear in the offsprings in the next generation, when a father non-rolling and a mother tongue rolling can be crossed. So here it is an example of tongue rolling, inheritance of tongue rolling, and you can make this type of pedigree chart for different genotypic features which expresses itself in F1 generation or in the daughter, in the offsprings. Now Punnett square. Punnett square is the structure in which in a square 4 subsquares for monohybrid cross and 16 subsquares are present in a big square for dihybrid cross. So here Punnett square, it is first proposed by Punnett, and therefore it is known as Punnett square. Here suppose it is a Punnett square. Here in a big square there are 4 subsquares. It is for monohybrid, study of monohybrid cross. Here we are writing gametes. Suppose here we have to write female gametes and in this side we have to write male gametes. After it, when these are crossed, then after crossing genotypes are written in the subsquares as well as we have to write phenotype. For example, we are taking an example of hemophilia or color blindness. Suppose here color blindness. It is an example. Color blindness is the genetic disorder in which person can't identify red and green color mostly. So it is a genetic disorder. Here two types of conditions may take place because it is a disease which are known as X-linked inheritance. So here we are writing an example. Here these are the female gametes. If the female is carrier, then she may have here degree means here the gene which is located on the X chromosome is expressed. So X degree and X, these are the two gametes which are known as female gametes, and here we have written XY which are normal gametes. Now after making a cross between these are the four different types of zygote appear in first generation. These are the genotypes, and now we have to write phenotype. Here it is carrier daughter. Here it is normal daughter. Here it is color blind son, and this one is normal son. So in this way, Punnett Square help in study of any type of genetic disorders or inheritance in a family when a male and female are crossed with each other. Here these are the four offsprings. Here genetic combinations means these are the genotypes. These are genotypes and these are the phenotypes. So Punnett square is the square in which 4 or 16 subsquares present, and it is used to study about the inheritance or inheritance of genetic disorder in a family. So here we have to write female gametes, and on second side we have to write male gametes. Then after crossing we have to write in each of the box or in each of the subsquare at first genotype and then phenotype, which are expressed by these genotypes. So it is a Punnett square. So in the next video we will study about sex-linked inheritance and a number of examples for them. So don't forget to watch the next video because it may be very helpful to you to understand how inheritance of genetic disorders can take place. There are two different types of inheritance, X-linked and Y-linked inheritance, and these are collectively known as sex-linked inheritance. So in the next class we will discuss about sex-linked inheritance. Thank you, students.